Okay, let's get started. It's 12:01. We have about 18 people, so we have around 20, so we're doing well. So it is my great pleasure to introduce Jameson Blount. Blount? Did I say that right?
Blunt, but I don't blame you
Blunt.
for it's
Jameson
spelled
Blount.
Blount. Yeah.
Okay, Jameson Blount. So he was invited to speak today by Piotr Prince, who's not here. So I will give a brief introduction. So Jameson is a fourth year graduate student in computational biology and bioinformatics at Duke University. His primary mentor is Dr. Ryan Baugh, and Jameson also received a Master's of Biostatistics at Duke in May of 2021 and is also a graduate of the University of North Carolina, Chapel Hill. So we were talking with Jameson and the grad students this morning, and Jameson has been very interested in developing novel approaches for GWAS and complex trait variation. He's working in C. elegans as his model organism, and sort of his research more broadly recently has been focused on understanding the genetic basis of starvation, resistance, and phenotypic plasticity. So also I think Jameson would like to give a little bit of a explanation about how he got here through his interactions with Piotr, who could not attend today. So
Thank
take
you
it
for
away,
that.
Jameson.
Oh, yeah, I will. Thank you for that introduction. I appreciate it. Right. So as you can see by that introduction and the title of my talk, I'm interested in moving statistical genetics methodology forward, whether that means writing those updates into software or changing how people use existing methods. And that is how I met Dr. Piotr Prinz, who invited me here to speak today. Among his many projects, Dr. Prinz is a primary contributor to the genome-wide association software called GEMMA. Early in my PhD, I started poking around in its source code, and I started bothering him over email with a lot of questions. And so he and I have been working together ever since on revitalizing GEMMA as a part of my thesis work. The other part of that work has to do with the similar genome-wide association methodologies that we use for model organisms, C. elegans especially. It's a problem that sort of arose from analyzing our own complex trait data, as I will get into throughout this presentation. So before I begin with the presentation in earnest, I would also like to acknowledge some other very important people, my advisors, Andrew Allen and Ryan Baugh, my thesis committee members, Paul McGuiney, Greg Ray, and Tim Reddy, as well as the rest of the Baugh Lab for their support and contribution throughout my work and throughout my PhD. So genome-wide association studies began with humans and historically have had a lot of success in isolating individual genes that are involved in disease pathogenesis in cases like macular degeneration and cystic fibrosis. However, as GWA has been used to explore more and more traits and phenotypes, researchers have learned that some phenotypes are much more complicated than those diseases that I mentioned earlier and can be better investigated using model organisms. Trait mapping within model organisms can identify the genetic basis of complex traits by analyzing phenotypic variations across large populations. This process can provide insight into human diseases where direct experimentation is not feasible. Due to their specific strengths, these model organisms each have their strongest impacts in different areas of biology. Insulin signaling and starvation resistance are examples of polygenic traits that cannot be measured reliably or ethically in humans, but they are traits that can be well examined within the nematode roundworm Caenorhabditis elegans. C. elegans offers a particularly versatile model for the genomic study of environmental stressors and metabolism, since the animal is frequently starved in the wild and has robust, well-characterized starvation responses. C. elegans also has minimal requirements for growth, the ability to produce large numbers of offspring through self-fertilization, and a compact, well-studied genome. Its life cycle, pictured here, is relatively short and predictable, with a generation time of 3 days when conditions are stable, and its environment can be tightly controlled. And since C. elegans is also transparent under a microscope, this allows us to observe the effects of our genetic experiments at a cellular level and across generations. So we really like working with it for what we have in mind. When the larva worm's food source is depleted and the animals are crowded, C. elegans larvae activate an alternate life cycle and molt into a larval stage called the dower, seen at the bottom here. This morphological transition is initiated by the anticipation of starvation. They can live in this state of arrest for up to 4 months or until they encounter more favorable conditions, at which point they will resume development as a fourth stage larva or L4. But C. elegans can also reversibly arrest development earlier, during the first larval stage in response to hatching without food, pictured here on the right of the slide. L1 arrest, also known as L1 diapause, occurs without the morphological modification of the dower, but is accompanied by a similar increased stress resistance. We at the Ball Lab are interested in the signaling and gene regulatory mechanisms that enable development of an entire animal to stop and start in response to nutrient availability. We often focus our study on the L1 larvae because fed L1s can act as a natural genomic control for arrested L1s because of their physical similarity. It allows us to investigate the molecular mechanisms that enable arrested larvae to endure starvation. Despite the evolutionary distance between us, C. elegans shares humanity's propensity for globetrotting, as you can see here. This map is from the Caenorhabditis Natural Diversity Resource, which is a publicly available resource for genomic mapping data and tools, such as interactive genome tracks, heritability calculators, and a phenotype database for 3 different species of Caenorhabditis. That's Caenorhabditis elegans, briggsii, and tropicalis. Each of these red pins on this map represents a location where a diverse wild strain of C. elegans was gathered in the field. So there's a lot of natural variation in how these animals handle starvation, just like in humans. But like all model organisms, C. elegans have unique population genetic quirks that are not always accounted for in trait mapping, and this complicate the use of standard platforms for statistical inference. Excuse me. C. elegans are primarily hermaphroditic in the wild, and so while males also do exist in the wild, they're rare, meaning that C. elegans more often reproduces through self-fertilization. They've also experienced bottlenecks in their recent evolutionary history that exacerbates inbreeding within the system. This unaccounted differences in population genetics highlights an urgent need to adapt human-based genetic tools for use in model organisms in order to successfully map and study polygenic traits. One especially problematic consequence of C. elegans' unique biology is that extensive regions of their genome often occur together non-randomly. The measure for this non-random association of alleles is referred to as linkage disequilibrium, or LD, and it most often occurs between loci that are in close physical proximity. However, when it comes to C. elegans, this association is not limited to physical proximity because there is LD both within and between chromosomes. To give you a better idea of just how pervasive LD is within the C. elegans genome, we can compare their rate of LD decay to humans. This metric demonstrates how quickly LD decreases as a function of physical genetic distance. So on the left side of this slide, there are 2 plots from a study of linkage disequilibrium within human chromosome 22 by the Human Polymorphism Study Center in Paris. In the top figure, each dot represents the Pearson correlation coefficient, or R squared, between pairs of SNPs as a function of the distance between each member of that pair of SNPs. You'll notice that the maximum R squared between any pair of SNPs is less than 0.2 when the SNPs are 1,000 kb from one another. So on the far right of that panel B. The bottom figure shows the average LD decay within human chromosome 22 within other human populations as well. So pictured in red is the original Human Polymorphism Study Center. The green line represents an unrelated UK sample. The blue line represents a combined UK and Human Polymorphism Study Center population. And the violet line represents an Estonian population. In all populations, the average R-squared values are less than 0.05 when the SNPs are as close as 400 kilobases. On the right side, we can compare these trends in humans to the average LD decay in all C. elegans chromosomes. C. elegans have 5 autosomal chromosomes and 1 sex chromosome, chromosome X. Again, each dot here represents the R-squared values between pairs of SNPs as a function of their physical distance from one another. And the solid lines show the averages across all SNPs on a given chromosome. I want to draw your attention to 2 things on this plot. First, the scale of our physical distance is now on the order of megabases. So at 1 megabase, which is the equivalent of 1,000 kb, that was seen on the earlier slide, we're looking at R-squared values of around 0.4, more than twice the R-squared values of even the maximum R-squared between any given pair of SNPs that we saw on human chromosome 22. Secondly, I want to draw your attention to the SNPs that are perfectly correlated or have an R-squared of 1. Notice the sheer number of SNPs that are up there and the huge distance between pairs of SNPs that are in perfect LD with one another. In C. elegans, SNPs that are up to 13 megabases from one another can be perfectly correlated. That is a huge distance to see a perfect association compared to humans. GWAS tools were built with the picture on the left in mind, but most C. elegans researchers are taking those models and tools and using them for trait mapping within populations of organisms that look like what we see on the right. So it's clear that there are big differences that need to be accounted for. One of my thesis committee members, Paul McGuinness, and other authors sought to understand how frequently SNPs are in perfect LD within a few model organisms. They refer to SNPs that are in perfect LD as being genetically indistinguishable from one another because the configuration of alleles across the marker panel being tested are identical. So analyzing a single SNP of a genetically indistinguishable pair would give you the same information as if you had analyzed both. So on panel B, we have the median number of genetically indistinguishable SNPs as a function of the minor allele frequency for all possible allelic configurations at that given minor allele frequency. On panel C, we see the maximum number of GI SNPs across all allelic configurations, also as a function of minor allele frequency. You'll notice that there are 3 lines being plotted here, those for a data set of Saccharomyces cerevisiae, Drosophila melanogaster in blue, and Arabidopsis solania in green. The prevalence of genetically indistinguishable SNPs decreases as minor allele frequency increases. That is because as the minor allele frequency increases, the number of possible allelic configurations increases drastically. So the chance of an identical allelic configuration becomes very, very small. However, what panel C tells us is that even at fairly high minor allele frequencies, we're talking about potentially thousands of SNPs that are in perfect LD. Now, C. elegans is not pictured here, but it might as well be the poster child of the maximum number of genetically indistinguishable SNPs that you could see. And this is an issue for genetic mapping and genome-wide association because if alleles at a causal site and at one or more physically distant loci were to form a cluster of SNPs that are in perfect LD, it would be impossible to localize the causal site using just marker trait association. So to give you a picture of how that manifests itself in GWA and how LD impacts that, here's a preview of my work that I'll discuss later. What I'm showing you here is a Manhattan plot where the -log10 p-value is plotted for each SNP across the chromosome by its position. There are 2 significance thresholds pictured as the 2 dotted horizontal lines, a more conservative Bonferroni correction threshold in blue and a less stringent correction threshold in red. These are corrections for multiple testing since GWAS is often testing thousands of markers. So these are simulated results. And again, I'll return to this later, but in this simulation, I supposedly had full control of where the causal marker was for the given trait data. That causal locus was, of course, in the center of chromosome 2. You'll notice that there is what we call a quantitative trait loci, or QTL, that passes both significance thresholds in the center of chromosome 2. But there is also an additional QTL on the right side of chromosome 1. This demonstrates that even when the results should have detected only a single causal locus, more than one can crop up because of LD that occurs across chromosomes. And this is the problem we're dealing with. So we also want to know, how often does LD interfere with our mapping efforts within C. elegans? But to understand that, and to understand how we're approaching that, and where we can approach that within the model itself, this is a graphical representation of the mixed effects model that underlie most genome-wide association models. Unfortunately, there's not a lot of room within the model itself to adjust for LD. So when I say this is a mixed effect model, I'm referring to the fact that there are fixed effects, in this case, the genotypes, the genotype markers, and a random effect, pictured here by the genetic relatedness matrix, that essentially seeks to model how related each individual in your sample is to all other individuals in your sample. If we were just to use a simple linear model without the random effect, that would be assuming that the genotypes alone are responsible for the variation that we see in the phenotype. Our use of the mixed effect model structure is an acknowledgement that there is a component besides the genotypes alone, in this case, the relatedness between individuals, that are responsible for the correlation between individuals' phenotypes. Especially in C. elegans, we use the mixed effect model to control for the relatedness between individuals. And so to identify a causal marker, this model attempts to identify how much of the variation that we observe in the phenotypes is due to genetic versus non-genetic sources. The genetic sources being, of course, the marker genotypes and the genetic relatedness. What isn't genetic is attributed to what's called the observational noise, or the residual variance component, pictured in the VE. And those two components, the genetic variance component and the residual variance component, together make up the total variance that we see observe in the phenotype. Now, like I said, there isn't really anything present in this model that could really account for the extent of the LD problem that we see. And that's what we're working on. And no one else in the field of C. elegans specific statistical genetics is working on this problem. So our contribution to the solution is that we are trying to come up with new ways to account for these quirks and trait mapping at several different places. So we're using statistical fine mapping and genome-wide patterns of linkage disequilibrium to try and inform our GWAR results. And we're also working on a method that harnesses the typically ignored information within biological replicates. So the first challenge in getting the full landscape of linkage disequilibrium is a computational one. Even if you're working with a subset of all the wild strains, the number of pairwise comparisons needed for the SNP panel is usually computationally intractable, since the number of pairs that you need to compute scales exponentially with the number of SNPs that you're working with. Well, we can actually use extensive LD to our advantage here. Like I mentioned earlier, SNPs that are in close physical proximity are often in perfect LD. So choosing to analyze one of a pair of SNPs in perfect LD gives you the same information as if you would analyze both. So if we extend this logic to blocks of SNPs that are in perfect LD, we can again get the same information from that block by pruning that block down to a single representative or tagging SNP. If we do this for all blocks on all chromosomes, plus filter for things like missingness and minor allele frequency, which are usually filtered out of SNP marker sets for GWAS anyway, we go from having to analyze all pairwise comparisons for 750,000 SNPs to only having to look at 21,000 21,000 SNPs and every pairwise comparison that comes with that. And that's a much more doable number. So this is a snapshot of an R-Shiny app that I've developed to better visualize the landscape of linkage disequilibrium for those 20,000 SNPs within the C. elegans genome. Now, this is a snapshot in more than one way, because the patterns seen here reflect a specific population of 192 wild strains, and only a subset of all the possible SNPs that could be plotted are plotted here. Note also that what's plotted here are the R values, not the R squared. So we can visualize not only the strength of the association, but also the direction of the correlation, so positive and negative correlations. Excuse me. As you can see, there is not only substantial correlation within chromosomes, as is expected with linkage disequilibrium, but there are swatches of this fabric that have high LD between chromosomes, in particular between chromosomes 4, 5, and X. Now, let's say I wanted to investigate one of those patches of high LD. You can zoom into those hotspots with this R-Shiny app, and I've done so for a hotspot that is between chromosomes 5 and X. As you look at this, you can see individual SNPs that are sort of responsible across all other SNPs for these sort of hotspots. And as a part of this R-Shiny app, you can export the data table for this submatrix if you need to examine those SNPs in follow-up, or just have an idea of what the actual values underlying these are. To give you an idea, in this particular area, the peak association between SNPs on different chromosomes were around 0.8, and that's really high. So what are we going to do with this new information? We are using these genome-wide patterns of linkage disequilibrium to perform statistical fine mapping with the sum of single effects model, or SUSY. This is a Bayesian framework for fine mapping. So the process involves taking GWAS summary statistics, which represents our prior beliefs on which loci is associated with the phenotype data, and update those beliefs using the genome-wide patterns of LD that we've just calculated. What we get from this are posterior inclusion probabilities on credible sets of variance. Again, in a Bayesian framework, it's this informed posterior distribution that describes the relative support for the association of combinations of variance. It's through this statistical fine mapping that we can deconvolve the noise that extensive LD creates in association signals and narrow our focus down to the variants that are most likely responsible for the associations that we see in GWA. So in order to test this pipeline and see just how effective it is, I've already run a few simulations. But first, I'd like to discuss how I generated the phenotypes for these simulations, because it's very important for the quality of our sort of quality control, our analysis. So we can stress test this framework. So I'm using the same linear mixed effect model that the GWA expects from the phenotype data. But the dials that I can turn and test revolve around the sources of variation within the model. So heritability and effect size are my dials for the genetic variance. And I can also control how much observational noise is in the data. And that's my dial for the residual variance. But in addition to these dials, I can control which variant or variants are causal, changing the underlying architecture of the trait to be either simply monogenic or more complex and polygenic. And where these causal SNPs are within the genome-wide landscape of LD also matters for our mapping power. So the choice of which SNPs to make causal matters as well. For each combination of parameters, so the heritability, the effect size, the number of causal QTL, what does the local and trans LD look like for a SNP, and the minor low frequency, I simulate 100 phenotypes to get at the average behavior of a particular parameter set. So here's that figure I showed you earlier, where I analyzed the tagging set of 20,000 SNPs. For the trait data that went into this, I simulated that SNP in the middle of chromosome 2 to be causal, with a heritability of 0.25 and an effect size of 0.5. In addition, you can now see the posterior inclusion probabilities for the SNPs that fell within the QTL boundaries on chromosome 1 and 2. These QTL boundaries are called based on a heuristic that establishes a confidence interval of predetermined length on either side of the peak SNP based on this association signal. You can see here that SUSY is able to reduce the signal on the other QTL that came up on chromosome 1. It has no faith in any association being significant on that chromosome. While the SNP that was at the peak of chromosome 2 is the only SNP with a high probability of being the causal SNP. Clearly, the association noise has been reduced here. I will note, though, that the SNP that's listed to be causal here, the 7882973, it's actually not included in the sampled SNP set, even though it was simulated to be causal. This was an entirely conscious choice on my part, because the SNP that is causal is rarely in the marker panel that is being tested in the real world. So this was our way of making our test of this pipeline a little bit more realistic. The peak SNP in terms of significance is actually about 16 kilobases away from the causal SNP. It's seen in the peak SNP on the left, and it's the red dot SNP on the right. This SNP is not in perfect LD with the causal SNP. However, the two SNPs, this SNP here and the one that was left out, have an R-squared correlation coefficient of 0.57. So it makes sense that in searching for the association signal of the causal SNP, it picked this one up. The only other SNP that's in perfect LD is actually not tested in this set either. So what happens if we decrease the heritability? So it's the same set of SNPs being tested, same everything, except the heritability has been decreased. You'll notice that the association signal has definitely suffered from this. That middle chromosome 2 peak is no longer nearly as high. It's actually less than the peak that we saw in chromosome 1. And because of this, you'll notice that Susie is a lot less confident in that SNP being causal as it was in the previous phenotype. But that's because it was given a relatively weak prior, if we're still keeping the vernacular of Bayesian analysis. These GWAS, again, are acting as the prior. And if we give it a weak prior, it's not going to be able to do very well. It has a harder time finding the signal from the noise with a weaker signal overall. There's less that it can do with it. What happens if we change the location? So here, I've changed the location to be on the left side of chromosome 5. And I chose this site because there's a lot more LD between different chromosomes. And this is evidenced by the fact that there are QTL that are cropping up all over the place. We've got one on chromosome 1, 2, and X. And so even though that signal on chromosome 5 is really, really strong, that gave Susie a little bit more pause because, like I mentioned, that association signal is actually much stronger than what we saw for the chromosome 2 QTL. But Susie is still a little less sure about it. Its probability is in fact above 0.5, but not much above 0.5. So the fact that that association signal was diluted across other chromosomes does sort of have an impact. But it's still remarkable that Susie is able to pare down on all that other noise so completely. These results are really encouraging. But again, this is evidence that the surrounding landscape of the causal SNP is really important to consider. So we're doing this for a total of 8 sites that represent different combinations of low and high LD, whether that LD is acting in cis or trans, and of course the minor allele frequency of that causal SNP. But when we're doing all this, there needs to be a metric that measures the fine mapping performance quantitatively. So we're not just eyeballing the before and after each iteration. We can do this by measuring how close the most likely causal variant is to the true causal variant before and after the process of fine mapping, and see how often Susie decreases, increases, or does not change the distance to the causal SNP. So this is a theoretical example. Let's say that the true causal SNP has a location that's that vertical dotted line. It's not in this set, I'm not testing it, but that's its location. And let's say that I ran GWAS on the marker set for a given phenotype, and this is the results that it gave me. This SNP that's a little bit farther, or actually a lot farther from the causal SNP, actually has the highest association signal. That distance from that SNP, the most significant SNP according to GWAS, to the location of the causal SNP is our pre-Susie distance. And then once we do fine mapping, let's say we actually get, Susie is able to figure out that, oh, okay, this SNP received more association signal, but it's actually the one that's closer to the causal SNP that should be the most likely causal variant. And so the distance from that SNP to the true causal variant's location is the post-Susie distance. And so what we're looking at here is how big is this difference between the pre-Susie distance and the post-SUSY distance. Here is how I've chosen to display this performance. The distance from the most significant SNP in GWAS to the true causal SNP for a given iteration is plotted on the x-axis as the pre-SUSY distance, while the distance from the SNP with the highest pip to the true causal SNP is plotted on the y-axis. I've plotted 3 heritabilities worth of data here. So there are close to 300 data points plotted, one for every case in which at least one SNP passed the genome-wide significance threshold, color-coded by the heritability of the phenotype. So entries along the diagonal dotted line show iterations where GWA and SUSY agreed on the causal SNP because they didn't change. Entries below the line show iterations where SUSY got closer to the causal SNP than GWAS did, since the pre-SUSY distance is larger than the post-SUSY distance. Entries above the line show iterations where SUSY got farther from the causal SNPs. There really isn't too much of a strong trend here, but it does seem like higher heritabilities are clustered around smaller distances overall. I want to draw your attention to these cases because they're actually cases in which the most likely causal SNP was on a different chromosome than it should have been. They're labeled infinite cases. So in this case, it was sort of hard to figure out a distance to assign to something that's on a completely different chromosome. So what I've elected to do instead is just assign an arbitrarily large distance to it. So the dots that are on the line are instances in which a most likely causal variant that was called on another chromosome stayed on another chromosome, where the dotted line, the dots on the bottom of the x equals y line represent cases in which SUSY actually brought the most likely causal SNP from a different chromosome onto the correct chromosome. So it did a great job at bringing it back to where it needed to be. And of course, those cases on the top represent cases in which SUSY actually said the causal SNP was on a different chromosome. So to put a number to what I was discussing earlier in terms of how well SUSY does, a majority of the time SUSY either maintains or improves GWA results, because 247 plus 24, that's the number of times that SUSY is able to either meet GWAS or improve it. And like we saw earlier, what may cause these 10 cases are instances in which there's just been a low association signal, and SUSY just wasn't really sure, because clearly what the signal that you give SUSY matters. We also are eager to apply this pipeline, once proven sound, to real data. As a way of teasing out the natural variation in starvation resistance, our lab came up with an assay that would allow us to use population cell selection and sequencing to characterize natural variation in starvation resistance. In this experimental setup, a population of 192 isotypes of wild strains are cultured in standard laboratory conditions, are pooled, and then subjected to starvation. You'll notice that this is that 192 that I've been talking about, and that was very intentional, because this pool of worms represents, theoretically, the right amount of genetic commonality in the population and genetic diversity. So this is why we're sticking with this set of 192 strains. We expected actual representation to vary across strains and replicates, so we collected DNA from an aliquot of L1 larvae from this pooled population as a baseline sample to capture initial population composition. In addition, aliquots are taken from the pool at days 11, 14, 17, and 20 of starvation. And sampled larvae are allowed to recover with food for 4 to 5 days, enabling reproduction for 1 day, and then the entire population is collected for DNA prep. DNA prep from baseline, as well as samples, are allowed to recover, reproduce following starvation. So we have 5 replicates of this. So what the phenotype data looks like for this experiment is pictured here. It can allow us to examine which strains are more resistant to starvation compared to those that are more sensitive to starvation. Pictured here are the top 25 and bottom 25 strains according to their trajectory survival over time. but we can zoom into just the top and bottom strains. So each black line is a trajectory for an individual replicate, where for each day, we're plotting the log2 day n frequency that's normalized to day 1. So we're trying to sort of get a representation of how much does this strain change to its own base relative to its own baseline. The blue line represents the average frequencies across replicates at each time point. What I want to draw your attention to here, which transitions me nicely into the next part of my thesis work, is the individual black lines. Biologists, especially those working with model organisms, conduct multi-replicates like this, but the trait and data values from each of these replicates are averaged across all replicates in order to generate a single trait value per individual for trait mapping. This variation between replicates often go unaccounted for, but it's really important to account for these, especially when your goal is to understand the impact of natural variation among populations of wild strains on trait expression. In a setting like that, there's the potential for significant variation between biological replicates within a strain and even between these strains. Averaging, which is what most people do, ignores the non-genetic experimental variance surrounding trait values and assumes that the observed variance in trait values is due to genetics alone. So oftentimes, when we're doing these kinds of analyses, we use ANOVA to compare the means of several different groups. ANOVA is actually a mixed effect model, or other way around. The mixed effect model that we are using for GWAS is a form of analysis of variance because we're comparing the differences that we observe between the within group variance and the between group variance. In this case, you can conceive of each strain as being a group, since we often have more than one measurement for a given strain. However, with most multi-replicate studies, we're actually not fully utilizing the principles of ANOVA when we run GWAS, because what we're doing is we are mapping each individual's mean without taking into account the variation within a group's mean. So in most GWAS models, this is what we're assuming. We're assuming that each individual has the same residual variance, regardless of whether that's actually true or whether we see that in the phenotype data. My modification to this would be to give each individual their own, or each strain in this case, its own residual variance based empirically on the trait data that we have. So in this case, we would be calculating the variation in the slope of the line across a strain's 5 replicates and incorporating that into the GWAS itself. So my expectations for modeling the individual specific variance is to create a better informed statistical model by directly specifying this residual variance component in the model. In doing so, this frees up the total variance in trait values and allow for more accurate inference of the genetic variance component. So this is the other project that I've been working with Piotr. The math itself involves a lot of sort of recomputing, because that assumption is really baked in to many statistical assays, and especially this model. So this has been a matter of sort of, it seems like a very minor change, and it is a minor change when you're just thinking of the theory of everything. However, in practice, it's been a lot more challenging, especially because I've had to learn C on the fly. So, in conclusion, a genome-wide map of linkage to disequilibrium provides the context for potential trans chromosome associations for past and future trait mapping experiments in finer detail. These modifications also integrate previously ignored information into the process of trait mapping for gains in statistical power. And our ability to map polygenic traits with increased precision will reduce the search space for follow-up genetic studies on follow-up for the causal variants of interest. Yeah, that's all I had. Thank you.
Thank you for that. Some applause. I see some applause in the audience. So we definitely have some time for questions, and you can either put them in the chat or raise your hand and we can give you the mic and unmute yourself.
Stop screen sharing now.